We're going to switch gears now and move into a discussion of energy. So far we've said that catabolic reactions release energy and anabolic reactions require energy or consume energy. And we've also talked about how enzymes are required for virtually all biological reactions to make those reactions occur more quickly. So, what exactly is energy? We've touched on this difficult topic a couple of times now in previous lectures. Remember, the traditional definition of energy is the ability to do work. Now, if we think about that definition on more of a biological level, how that definition applies to our microbes, we might think of it as meaning that energy is the ability to cause some kind of change in a cell. It might be a physical change. Perhaps we need to move something from one location in a cell to another. Or it might be a chemical change. We might need to, again, alter what kind of molecules are present in a cell. The important thing to remember is that all living things require a continuous source of energy. It doesn't matter if it's a microbial cell or a human cell or some other kind of cell. Energy is critical to the survival of that cell. Something else we've mentioned before is this first law of thermodynamics, and this is a good time for us to review it. The first law tells us that energy can neither be created nor destroyed, only altered in its form. Microbial cells cannot make energy out of thin air, and they can't destroy energy. All they can do with it is change its form. Microbial cells basically have a couple of options when they're faced with a metabolic reaction that releases energy, for example. They could choose to store that energy by building new covalent bonds. That's what we would refer to as potential energy at that point. And remember, inside that covalent bond, there's a pair of electrons. That's the stored energy. The electrons we think of as being the conduits of energy, the guardians of energy. So, energy can be stored in a microbial cell, or it can be transformed into some kind of energy that the cell has a need for at that moment. Perhaps it has a need for kinetic energy, that movement energy. Perhaps it has a need for chemical energy. Maybe it has a need for thermal energy. We can transform energy from one form to another, or, as we said, we can store it for later. Now, we've talked about how this molecule called ATP is a critical player in the dynamics of energy in a microbial cell. We've talked just a little bit about how ATP is made, about how cells use processes like cellular respiration and oxidative phosphorylation to make ATP. We've talked about the fact that microbial cells put a molecule called ATP synthase in their plasma membrane, and they use a proton gradient or a hydrogen ion gradient in the periplasmic space to use the ATP synthase enzyme to make ATP. But what I want to talk about right now is what the cell does with ATP. How exactly does a microbial cell use its ATP? And the answer is right at the top of this slide. Once ATP is made, ATP is used by continually breaking it apart and putting it together, and breaking it apart and putting it together. That's what the arrow represents here. When I say breaking it apart, I usually mean taking one phosphate group off of the end of that molecule. That's the pi. And we go from an ATP, a triphosphate, 3 phosphates, to an ADP, or a diphosphate, 2 phosphates. Every time an ATP has its outer phosphate broken off, a little bit of energy is released. And every time the phosphate group is added back on to ADP to make ATP, a little bit of energy is stored. So this molecule is used to store energy and then release that energy, and then store energy and then release that energy over and over and over again. Remember, I'm describing how microbes use their ATP. So when I say that a phosphate group gets added on to ADP to make ATP, this is describing how it's being used, not how it's being primarily produced by the cell. It's important for us to remember that living cells are very thrifty, and microbial cells are no exception. They are going to conserve energy wherever possible. And the way they do that is by making sure that any metabolic reaction that is releasing energy, they're going to harvest that energy. They're going to hold on to that energy. They don't want it to just be lost into the universe as heat. They're going to couple the exergonic reactions, the ones that release energy, with endergonic reactions, the ones that require energy. That makes sense, right? That's a thrifty way to manage your energy. Make sure that any reaction that releases it, you're going to grab that energy and then give it to reactions that require it. That's called coupling. And when a cell couples exergonic and endergonic reactions, in other words, catabolic and anabolic reactions, the energy is used as efficiently as possible. And that means as small amount as possible will be lost into the universe as heat. Now, unfortunately, anabolic and catabolic reactions do not always go on in a microbial cell at the exact same time. So it's not possible to directly couple anabolic and catabolic reactions. We need a middleman, and that's where the ATP comes in. Every time an exergonic reaction releases a little bit of energy, the cell is going to grab onto that energy by adding a phosphate group onto ADP to rebuild ATP. Now that little bit of energy that was released by that catabolic reaction has been held onto or stored temporarily in this molecule. Now the next time an endergonic reaction needs to occur, an anabolic reaction, a reaction that is going to build something and that requires a source of energy, the cell can turn around and break that phosphate group right back off of the ATP and release that little bit of stored energy. This process goes on again and again and again all day long in microbial cells. When there's an exergonic reaction, a catabolic reaction, the energy will be harvested by putting the phosphate group back onto ADP and making ATP. The energy is now stored here temporarily. Then when energy is needed for an anabolic reaction, it'll get broken off again and the energy will be released. This is how ATP is used. All right, we talk about the phosphate group being put on and taken off and put on and taken off. This is how a cell uses all of that ATP that it makes through things like cellular respiration and oxidative phosphorylation. This is just a diagram of this concept of energetic coupling, how a cell couples or brings together catabolic reactions and anabolic reactions so that energy is conserved. It's used as effectively as possible. And how the ATP molecule serves as a middleman. So if you start over here, the diagram shows us catabolic reactions are going on. Exergonic reactions are going on. In other words, energy is being released. If a cell did nothing right now, that energy would get lost as heat. The cell loses its ability to make use of that energy if it does nothing in that moment. So because cells are thrifty, they want to grab onto that released energy. They want to hold on to it. They want to save it. And they do that by sticking a phosphate group onto an ADP molecule and making ATP. Now they have stored that little bundle of energy that the catabolic reaction released. Now let's say the cell has a need to do an endergonic reaction, an anabolic reaction, something that requires energy, that consumes energy. Well, we've got energy right here. We've got a little bundle of energy right here. All we have to do is take the phosphate group back off, and that little bit of energy will get released. So ATP is just constantly rebuilt and then broken down, and then rebuilt and then broken down, so that it couples what's happening over here with what's happening over here. This molecule, ATP, is a really remarkable little molecule. It's not that complex structurally. This is a drawing of what it looks like structurally. Now, if you were to break this down, this is actually a sugar. This is what we call ribose. It's the same kind of sugar you find in RNA. Over here, this is actually one of the bases. This is adenine. Again, the A that you find in DNA and RNA. So far this looks a lot like a nucleic acid, but this is where it's different. Over here we see 3 phosphate groups. Remember, these are just, the phosphate group is just one type of functional group, or R group, that we talked about when we discussed biochemistry. A phosphate group is an interesting functional group because it happens to have a high energy associated with it. So this molecule has not 1, not 2, but 3 phosphate groups on it. And again, it serves as a middleman in the workings of energy in a living cell. This is ATP because it has 3 phosphate groups on it. When a cell needs a little bit of energy, it can break this bond right here, release this outer phosphate group, and now we have ADP. There's 2 phosphates left, and this outer phosphate group is off on its own. That's what we're looking at in this chemical formula. ATP is broken into ADP plus 1 phosphate group released. When the cell does that, when it breaks that outer phosphate group off, it gets 7.3 kilocalories per mole released. Remember, when you see kilocalories, that's what we call a calorie in layman's terms. So just a little bit of energy, 7.3 calories released, that the cell can then do work with. This molecule is so critical. This molecule is so critical to life that there are no cells on the planet Earth that don't use this molecule for energy purposes. In other words, there is no life without ATP. So, cells use ATP by taking off that 3rd phosphate group and then putting it back on, and then taking it off and then putting it back on. We also need to talk about how cells make their ATP. And this is where we get into that very complex series of metabolic reactions known as cellular respiration and oxidative phosphorylation, and so on and so on. So remember, when we think about eukaryotic cells, like human cells, cells that have organelles in them, the making of ATP is occurring in the mitochondria. This is where cellular respiration occurs. Cellular respiration is a series of metabolic reactions, not just one. and it's followed by a mechanism called oxidative phosphorylation, which is what actually makes the ATP. This is where ATP synthase comes in, which is located in the inner mitochondrial membrane in eukaryotic cells. Now, what about our prokaryotic cells? What about our microbes? We're not going to get into the fine details of cellular respiration and oxidative phosphorylation today, but I do want us to recall that in the cell membrane of a prokaryotic cell, this is where the making of ATP happens. There are some aerobic microbes that are doing cellular respiration, and there are anaerobic microbes that are doing cellular respiration. They do it a little bit differently, but the idea is the same. They're creating ATP through a long series of metabolic reactions. And you'll see I have terms like substrate level and oxidative phosphorylation here. Again, we won't get into the details of those today. I just want you to remember that in a prokaryotic cell, all of this is happening at the cell membrane because there are no mitochondria. I also want to just briefly mention today that there is a third way to make ATP, a third type of phosphorylation reaction. It's called photophosphorylation. I'm just going to mention it today. We're not going to get into the details of this at all, but I do want you to know that there is this third way for our photosynthetic microbes. So, if we put it all into a list, ATP can be produced in microbial cells during 4 different types of reactions. Oxidative phosphorylation is the one we talk about the most. This is the one that follows cellular respiration. This is a very efficient way for a microbial cell to make ATP. In other words, it gets the most bang for its buck. It gets the most ATP produced as easily as possible. ATP is produced in oxidative phosphorylation using a proton motive force, using a hydrogen ion gradient across the cell membrane. So the hydrogen ions, remember, are built up in the periplasmic space, and they're going to flow back into the cell through ATP synthase and create ATP in the process. What seems very strange to us is this. Oxidative phosphorylation can be either aerobic, requiring oxygen, or anaerobic, not requiring oxygen. Students find this very confusing because we want to think that this word oxidative means oxygen is involved somehow, but that's actually not what it means. Oxidative refers to oxidation reduction reactions, or what we call redox reactions, and we're going to talk about those in some detail. So, oxidative phosphorylation can require oxygen, or it can require something in place of oxygen. Anaerobic microbes do cellular respiration and oxidative phosphorylation. They just do it with something other than oxygen. Things like nitrate and sulfate and carbonate. Those are some terms that you should be familiar with. Substrate level phosphorylation is another way that ATP gets made in microbial cells, but it's not a great way for a microbe to do it because it's less efficient. Basically, what happens during substrate level phosphorylation is a little bit of ATP gets made by piggybacking the making of ATP on top of enzyme catalysis. And I'll show you a diagram of this in just a minute. I mentioned photophosphorylation. We're not going to talk about this one. I've got it in parentheses here. The reason we don't talk about photophosphorylation is because Even though this is a way that a cell is making ATP, a photosynthetic cell is making ATP, it's not making that ATP to use it for energy. Let me say that again, because that's a really important point. Yes, there are microbes, photosynthetic microbes, that make ATP using a process called photophosphorylation. The reason we don't think about it, though, when we talk about how microbes make ATP is because the ATP that's made during photophosphorylation is not used for energy. Instead, it is fed directly in to the making of carbohydrate. Remember, that's what photosynthesis is really all about. It's a series of reactions that allows a cell to make its own carbohydrate. So, again, I'd like you to be familiar with the term, but we're not going to talk about it because it's not a way that a microbial cell makes ATP for energy. Last on our list is fermentation. Remember, fermentation can be very inefficient depending on the cell, or it can actually be quite efficient. Some microbial species do fermentation only when it's absolutely necessary. It's a secondary metabolic pathway. They would much rather do something else, but when they can't, they will ferment. Other microbes use fermentation equally well with other metabolic processes. And some microbes are purely fermentative. It's the one they use as their primary metabolic pathway for making ATP. Remember, microbes are very diverse in their metabolic abilities. So to finish up this part 1 of microbial metabolism, let's take a look at this slide. I mentioned to you that there is a way to make ATP called substrate level phosphorylation. Substrate level phosphorylation is not a particularly efficient way to do it. You don't get as much ATP as you do if you use oxidative phosphorylation, but it still allows a microbe to make ATP. Substrate level phosphorylation got its name because it's a process that piggybacks onto normal enzyme catalysis. Take a look at this drawing. The green represents an enzyme. And as you can see, there is a visible active site here in this enzyme, as there are in all enzymes. Now, what makes this enzyme a little bit different is that in addition to having a place for the substrate to bind, and that's what you're looking at here, there's a second place where ADP can bind. Now, notice that there's a yellow ball drawn on the end of this substrate molecule. This yellow ball represents a phosphate group. When this enzyme is bound to its substrate, remember that's going to cause it to change shape and make some product. If we move over here to the second part of the diagram, the product molecule, as it's being made, is going to let go of that little yellow ball, that phosphate group, and it's going to be attached to the ADP to make ATP. So what we're seeing is this product is being released and ATP has been made along the way. The production of ATP piggybacks onto the production of product that this enzyme does. This is substrate level phosphorylation.